as we're motoring on to the finish now, we are very delighted to have, as Jim Sputich said, Mr. Laser Trap Single Molecule Person, otherwise known as Steve Block, as our final speaker this afternoon. Steve did his undergraduate work at Oxford University in England in physics. He did his PhD thesis work at Caltech on bacterial chemotaxis and how force generation is achieved by the flagellar motor with Howard Berg, who is here today. He then went to Stanford to do a postdoc with Jim Sputich, and he has since served on the faculty of Harvard and Princeton, and since 1999, Stanford University. His pioneering work on the use of optical tweezers, which I'm sure we'll hear a lot about today, has been recognized in many ways, including more recently by his 2007 election to the National Academy of Sciences. I should also note that Steve is an accomplished bluegrass musician. Wikipedia is just wonderful. But wait, there's more. He's also a member of a small and very select group of young scientists assembled to advise the US government on various issues of science and technology. This is the group called JASON. The name has been suggested to be an acronym of one sort or another. One suggestion was JASON stands for July, August, September, October, and November, the months in which this group typically meets. Also, someone has suggested it stands for Junior Achievers, Somewhat Older Now. In any case, this has led to some interesting papers on his CV, such as Block, SM, Living Nightmares, Biological Warfare Threats Enabled by Molecular Biology. So we all should feel a little bit guilty now. And you should also take a look. There are other interesting papers on his CV, which some of you who have posters today or occasionally present posters, do's and don'ts of poster presentation. Were you on the poster judging group? Okay, so we're going to see the... Not for today. Not for today, okay. And then one of my favorite titles, Block, SM, 50 Ways to Love Your Lever, Myosin Motor. So thank you very much, Steve.
Thanks. Thanks, I think, for that introduction. They say it's always intimidating to come to Harvard to give a talk. This is especially intimidating for me today because, as you heard it partly in the introduction, by an alignment of the spheres, it so turns out that in the audience today is my thesis advisor. I'm indebted to Howard Berg for that. My postdoctoral advisor, I'm indebted to Jim Sputich. My first collaborator as an independent investigator was Larry Goldstein, who gave a talk. And my collaborator of longest standing, Jeff Gellis of Brandeis University, is also here. And I won't out them, but there are actually some people who serve on JASON in the audience as well. So for all those reasons, I'm intimidated as I speak to you. I'm going to tell you at sort of at many levels in this talk about some of the problems we've been working on in the world of kinesin function. You see an example of a kinesin motor here in the first slide. As a matter of fact, that's a kinesin. And as you heard from Larry, who gave a wonderful introduction to this, it moves vesicles along microtubules. You also heard from Jim Sputich about two-headed motors that consume ATP and move, and kinesin is an example of that. So in many ways, some of the background has been done for me, and that's going to turn out to be very helpful. I'm going to exalt the field of single molecule biophysics for you here today, and you may ask, why are we so interested in it? Well, if your name is Stephen Michael Block, it behooves you to work in this area. Here is the gel, as it says here, the workhorse of biochemistry. For many years, this is the sort of bulk assay that people have been using to resolve, for example, various things. In this case, what you're seeing is RNA being made by an enzyme RNA polymerase, and these are the short ones are down here, the long ones up here. And as time goes on, the RNAs get longer and longer. And one thing you notice is that some of these bands, which correspond to single base pair positions, are dark. Some of them are light. And that's because there's a phenomenon called polymerase pausing. So it pauses at these places. But from data like this, you can't tell whether a lot of the polymerases have paused for a short period of time, or a subfraction of the polymerase is paused for a long period of time, because what you get here is something that resembles the probability of pausing times the duration of pausing. The darkness of the band is proportional to that fraction. And what you'd really like to do is get more information. This is akin, by the way, to looking at something like the Boston Marathon from an altitude of several hundred feet, and maybe you can make out the water stations here where there's an agglomeration of people. What you'd really like to do is study someone like this. There's Robert Chariot, 4-time winner of the Boston Marathon. And so this is the task we have before us in the area of single molecule biophysics. Wouldn't it be great if we could study these molecules one at a time and measure their magnificent motor-like properties? And of course, you've been hearing today a number of experiments that take you along that direction. This, just by the way, is just to prove that it can be done. This is that same molecule of RNA polymerase we were talking about before, but now a single molecule transcribing along a single DNA template. And what you can see from this record, and you'll hear about how this, broadly speaking, how this is done, but from the single record, you can see that it moves at more or less constant speeds, interrupted by a series of pauses. But now you're resolving pauses at the individual molecular level and not through bands. And here you can determine independently the probability of pausing and the duration of pausing. And you have all those numbers from data like this that you would not be able to get from a gel. So I'm here to tell you more about kinesin. I've been indebted to Larry for a wonderful introduction to this. And in fact, in his introduction, he talked about that neuron that's in your spine that goes down to your big toe and the problem of how you get organelles all the way out there. So you're very familiar with this problem already. And as you know, in the axons of nerve cells are large numbers of microtubules. They move cargo. Here's an EM picture, and it's been color-coded to sort of correspond to this cartoon. There's a cargo. There's something that was purportedly a kinesin motor, and there is a microtubule. And this is supposed to move on that, carrying the cargo along. And you've already heard also from Larry about a number of diseases, including Alzheimer's disease. You heard about various types of disease in dyskinesias in cilia and so forth. And these things all can result from, in some cases, mutations in your kinesins. If you get smallpox or one of these horrible viruses, they actually hijack the cytoskeletal structure and they move viruses along it as cargo. And that's how certain types of viruses get from cell to cell in your body. So they've been subverted by viruses. They are problems in disease. They are targets, as you heard from Jim. He talked about the myosin targets for cardiomyopathies. Attractive anti-cancer targets are various types of kinesin molecules. So this is actually nature's tiniest mechanoenzyme that runs on ATP, at least as far as we know so far. And here's a video that was, to some things, to flights of fancy, but it was done right here at Harvard, and it's from something called the inner life of the cell. And there's a kinesin motor, and you can notice the way it's stepping. A lot of the properties that you see in this film, of course, the Brownian motion is absent, but the stepping properties of this motor have been worked out over a number of years using the kinds of experiments I'm going to tell you about today. And those properties where this thing moves hand over hand and it walks seemingly along a tightrope as this, carrying here Jabba the vesicle, blub, 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 behind it, specifically attaching the cargo to this thing. All those properties have been gleaned by a number of experiments that were conducted at a single molecule level, and so I'm going to tell you a bit more about those now. As you've seen already in the previous talks a number of these so-called phylogenetic trees, the trees of life. These are the many different types of kinesin. Just as there are a multitude of myosins, there are a multitude of kinesins. There are over 14 subtypes that have been identified so far. Here's kinesin 14, for example. Larry talked about kinesin 1, which is found in this part of the galaxy there on the tree. And they come in many flavors. There are ones that are naturally dimers. There are the ones that have four heads, these tetramers. There are monomers. There's some that go the wrong way, some that go towards the plus end of the microtubule, which Larry was kind enough to define for you, and some go towards the minus end of the microtubule. In the human genome, there are about 50 different kinesin genes. So in every DNA and every cell in your body, there's the code for that, and each cell will express a certain subset of those. They're involved in organelle transport. Kinesin relatives are also involved in the process of mitosis and meiosis. They're involved in building the primary cilia and the other cilia that you heard Susan Dutcher talk about, and so they're important proteins. So here's conventional kinesin, or kinesin 1. It consists of two heads, a common stalk. So these are made from two independent polypeptide chains. They wrap around each other. They have a tail. Other proteins, light chains so-called, bind to the tail. There are these two little necks which connect the heads to the stalk. They have this coil tail structure, a tail. And a crystal structure has been solved, at least for the head part of it. This is from the rat, from the work of Kozielski et al. And you can see the two different heads. They each bind ATP, so each of these is an ATP-dependent motor, and they're connected by these things called neck linkers, which hook them up to the stalk, which on this scale would go right through Radcliffe Yard. And here's a fun fact. Jim talked about myosin. There's myosin in green, and there's kinesin in yellow, and they're virtually superposable over the extent to which they coexist. In other words, you can think of kinesin as a wholly owned subset of myosin. They've been superposed here by the ATP. Now, they don't have the same amino acid sequence, but they're close structural homologs. We know lots about this, and we learn lots about how it works. And some of the data will come out today. Some of the data is for, you can find in textbooks. But as I said, this is an ATPase. It binds ATP. We know it uses this as basically its fuel. We know, for example, that the heads bind microtubules in a way that depends on the ATP state. And when ATP gets hydrolyzed, it turns out it's the ADP state in kinesin, which is loosely bound and comes away. And this is different from myosin. Myosin is sort of the other way around in its cycle. So kinesin and myosin differ in some interesting ways, and this has important repercussions. We know that the dimers, these two-headed beasts, step in increments of 8.2 nanometers. You'll see why in a bit. They can take about 100 steps a second, typically, and Larry found conditions under which they can go even faster than that in vivo. And typically, they go for about a second before letting go, so they take about 100 steps and then let go. We know that, that every time one of these steps takes place, a single molecule of ATP is hydrolyzed. So it has a very good fuel efficiency. It takes one ATP per step. And as you'll see in a moment, the walk is the so-called asymmetric hand-over-hand walk. And this is something that Jim Sputich alluded to in his talk. The technology we've used and other labs have used to study this system is the so-called optical tweezers, or single beam gradient force optical trap. It was invented by Arthur Ashkin at Bell Labs. It's the closest thing to a tractor beam that man has yet produced. Using an invisible beam of laser radiation, you can grab and manipulate particles under the microscope, produces forces on them, measure displacements with exquisite accuracy. Currently, we're achieving 1 angstrom level resolution, which is the diameter of a hydrogen atom. We can make that measurement 100 times per second in real time on one protein. And it works because it turns out small dielectric things like little glass beads or silica beads or latex beads, anything that's transparent sort of gets pulled into a trap. And when I say anything that's transparent, that even includes things like bacteria. So this is for Howard Berg. This was actually done at the Roland Institute many years ago when I was in Boston. These are E. coli swimming around. You can see their bodies. You don't see their flagella because they don't scatter enough light, but now we've got one in the optical trap, and we can force it to do a square dance. We control the vertical, we control the horizontal. This is all done in real time. You see the clock going around. I never tire of showing this film. I show it all the time. All these years, it's still fun. We'll control the thing in Z now. We're going to go down towards the cover slip, and you'll see other cells come into focus. Then we'll come back up again. The cell is more weakly trapped vertically than horizontally, so the cell, which is a big long cylinder, orients itself vertically in the trap. So you're looking sort of down the barrel, as it were, for the cell. Cell's swimming with its flagella and trying to get out, but it can't. And before the animal rights activists come after us, we will turn off the trap, and it will swim away of its own accord. Okay, so that's an optical trap. We don't actually use them in my lab anymore to study whole organisms. We do use them to study molecules, and there the idea is to hook something like a bead, which takes the place of the natural cargo, the organelle, and we hook these beads up to single kinesin molecules, and we can do that. If this were done to scale, the kinesin were this large, the bead would actually be the size of this gymnasium. So this is not drawn to scale. But then we shine light through this, and by scattering laser light off the bead, we can measure its displacement with exquisite accuracy, getting down to that 1 angstrom level I was telling you about. And so here's a video where we've actually optically trapped a bead which has a kinesin molecule on it. There's a microtubule, and we put it down, it starts to move. It's moving because it's being pulled by this kinesin. Then it gets sucked back into the trap and starts to move again. You're going to see the film loop. So we've captured the bead, we place it on a microtubule, and then it starts to go. So this is more of a demonstration how it works. In practice, what happens is we don't actually leave the trap on. We actually use the trap to follow the bead and exert a constant force on it. This is something called a force clamp. And the concept of this is really simple. The bead is moving as a function of time. It goes through some displacement. And you have a trap, and with a feedback loop, you examine this position, and you keep the trap a certain fixed distance behind it. And since the trap is a spring made out of light, what you're basically doing is you're stretching that spring to exert this constant force. And in fact, this is that constant force. This is a histogram, which shows that we can keep this force constant to within about 5%. For an arbitrary distance as kinesin moves along the microtubule and eventually lets go. And for the physicist types among you, it's always good to clamp the conjugate variable. If you're a neuroscientist and you want to measure the picoamperes of current that flow through a neuron, you make a voltage clamp. And if you're a biophysicist and you want to measure the displacements, you make a force clamp. And in general, you want to clamp the variable that's conjugate to the one you want to measure, and then you can measure the other one with high spatiotemporal accuracy. So here's a record of kinesin moving, and now at much higher resolution than you typically see, and now you're beginning to see these individual steps that it takes as it walks. Each of these is a Kinesin step. They're a variable duration in time, but they're always the same distance. And you can do that by looking at a power spectrum of this. There's a very sharp peak there at 8.2 nanometers, which is not so much a surprise when you look at the structure of a microtubule. Here is a microtubule, and what you can see is that the barrel staves, or the so-called protofilaments of the microtubule, there are 13 of them around in most microtubules. Some have 11, some have 10. Different microtubules have different structures. But typically, they run more or less parallel to the longitudinal axis here. And what kinesin is doing is stepping from one tubulin dimer to the next. Tubulin dimer being a blue and a pink subunit here. And that spacing turns out to be about 8.15 nanometers. So kinesin is moving in exact multiples of this because the binding sites of the microtubule are in exact multiples of this. But why does it move in along single protofilaments and not wander over at random over the surface lattice? And how do the two heads coordinate to move? How much force does it produce? Why does it go so fast? Lots of questions. We can begin to answer some of these questions, and I should tell you that a lot of these have already been answered in the literature. Jim Sputich in his talk made a distinction between so-called hand-over-hand motion, which is the kind you get on the monkey bars, and the inchworm motion. And for the real biologist experts among you, many of you know that an inchworm is not a worm, it's an insect, it's a caterpillar, but it nonetheless moves in a fashion pretty much like this. And the distinction between walking, as I'm doing right now, where I advance one foot in front of the other, that's a hand-over-hand walk, as opposed to inchworming or crabbing or moving like that. That's an inchworm type motion. And I can even do a spinning motion like this, which also involves a hand-over-hand motion but has a certain symmetry to it. So one can ask the question, which of these many modes does kinesin actually walk in? And there are a number of ways of answering that. One way it was answered was an experiment not from my lab, but this was from the work of Paul Sullivan and Ahmed Yildiz and Ron Vale. What they did, and Jim Sputich showed you the myosin version of this, is that, which was actually done first, which is to hook a single fluorophore onto one of these two heads and watch it move. And that's like hooking something onto one of my two legs. When I take a step, my leg advances by twice the distance that my core advances in my body. Or put differently, my body advances by half the distance of my stride every time I take a step. And so they actually measured these steps for a kinesin molecule and discovered that the answer was closer to 17 nanometers, which is closer to twice 8, which is actually about 16. Twice 8.2 is 16.4. And it's closer to that than to 8. And so that was the experiment. You can actually measure the displacements by something, kind of centroid tracking, which they call Fiona, fluorescence imaging with 1 nanometer accuracy. Well, almost 1 nanometer. They got 2 to 3, close enough. Now that allowed them to then rule out the possibility of an inchworm model, and they were left with a hand-over-hand model. They then appealed to an experiment that Jeff Gellis conducted here, which was a very intriguing experiment, which gave a very counterintuitive result. When kinesin moved, it doesn't rotate its stalk to a very large extent. Instead of the cargo spinning, which it would be happening like this, and that's the symmetric walk, it actually walks like you and I do, which we have a left step and a right step, and the left and right steps are different. And so they argued that it was a hand-over-hand model, and they argued it was an asymmetric hand-over-hand model on the basis of the Gellis result. All right, so a similar experiment is this one. In this case, instead of putting a fluorophore on it, you can put a bead, and with a piece of DNA, you can actually hook that bead to a single kinesin head, and then watch that move. And here are some records from this. And sure enough, it's moving now in 16 nanometer increments. You can do a histogram of that. And this actually brings you back rather accurately the 16.4, which is the spacing that you expect for double steps. And so, indeed, kinesin is moving in a hand-over-hand motion. Is it symmetric or asymmetric? That's another question. By the way, if you ask what kind of force you can produce, this is the so-called force-velocity curve. It's how fast you go as a function of load. And so we're pulling it harder and harder backwards. And at some point, it's going to stall out. And wild-type kinesin stalls out at somewhere around 6 to 8 piconewtons of load. Doesn't sound like much, but it's a lot if you're a little molecule. And this is a number comparable to what the best myosins could do on a good day. Kinesin's usually a little better. And it turns out if you hook up to one of the heads instead of the stalk, you only have one head powering the motion, and you get twice the displacement, and you get half the stall force, which makes sense because the product of the stall force and the displacement, which is an energy, is conserved. So the thing's using about the same amount of fuel, and it's going twice as far, but it can only go twice as far with half the force. So how do we know it's asymmetric? Well, you could appeal to a different experiment. It turns out under some conditions, you can get mutant kinesin molecules which limp. So if I shoot you in the foot, or you twist your ankle, you'll limp. And what's interesting about a limp is if you were actually doing an inchworm motion and I shot you in the foot, every single inchworm would be affected by that. But if you're stepping leg over leg or arm over arm or hand over hand, then every other one is affected. And so it turns out that in these particular mutants of kinesin, if you look at this, you look at it every odd numbered step in this particular record. It might be even in the next record because we don't know the phase, but every other step here has a long distribution, and the ones in between have a short distribution. There's about a 6-fold difference in the timing of the alternate steps. The only way you can generate a limping molecule is to have an asymmetric hand-over-hand model. This is just an exponential for those distributions, showing that they have different time constants. And so just to remind you that this only happens in these strange mutants, this is a wild-type kinesin which has a full-length tail, and here now the distribution of steps is exponentially distributed, but there's no difference statistically speaking when you look at lots of records between the left and with the alternate steps that are even and odd. So since we know it's limping, and since we know this can happen, it therefore means that kinesin has somehow figured out how to do hand-over-hand stepping the same way that we do our walking, which is weird because we have a left foot and a right foot, and kinesins have 2 left feet. Kinesins have 2 left feet because they're made out of identical heavy chains, and when you put these things together, you're going to wind up with 2, in principle, identical molecules with 2-fold symmetry. In the crystal, it's not quite 2-fold, it's broken a little bit, but they're pointing in different directions. This is a problem because if they're going to have the 2 heads come down and meet the microtubule, one of the heads has to spin around completely, which means that the microenvironment of one head is very different from the other. In this particular thing, the head-neck linker comes out, as it were, the front of the thing, and the head-neck linker here comes out the back of the thing. So somehow it breaks symmetry, and somehow it knows its left from its right. And how do you walk hand over hand like that and not have your core spin, as we learned from Jeff Gellis? Well, here's one view on how you kind of might do it. This is kind of Robokinesin. All the interesting physics has been removed from the problem. There's no Brownian motion. There's no never mind. So before you get upset, Howard, you know, this is supposed to be a low Reynolds number. But the point is just a topological point, which is that if you look carefully at the blue and green head, notice that it swings out of the plane of view towards you. And now fixate on the purple head and purple and red head. You notice it swings in the plane of the screen. And they have to move in these separate paths in order to keep their legs from being tangled here, and that's the only way they can do this in such a way as to keep this moving without having it rotate. Now, these are not the unique trajectories. You can imagine other trajectories, but the topological point here is that those trajectories have to be different. You can't have the same trajectory for both heads and not get all twisted up. The only way you can have it the same is if you do that rotation thing, and we know that kinesin doesn't do that. So this is getting interesting. Whereas it's true that kinesin in these mutants alternate in their timing between long and short, they don't actually alternate in their size. So if you measure the displacements of the steps rather than the time of the steps, which are these distances upwards, you discover that they're all the same. And that's kind of interesting because it means that the left steps, which we can separate out in the limping mutant, we couldn't do that in the wild type because we don't know what's left and what's right. But in this case, we can separate out one of the steps, call them left, I'm just using that for a name, even numbered subset, short duration step, doesn't matter what you call them. They have a displacement histogram which looks like 8.2 nanometers, and so does the other phase. The slow phase has the same. And here's the wild type kinesin, and of course its steps also have this. And the fact that all these steps are always the same allows us to make some statements about how kinesin must be walking. Because it turns out that if you're walking, for example, along 2 adjacent protofilaments like this of the microtubule lattice, because those filaments have a tilt with respect to one another, you wind up actually stepping the centroid of the molecule, which is shown here in yellow. It winds up instead of straddle move, winds up alternating between approximately 7 and approximately 9. So you get 7, 9, 7, 9, 7, 9. You don't do strictly 8, 8, 8, 8. And so of the many models that could have been entertained about whether you do this, it turns out you're only left with, when you think this through, these 2 models. And these 2 models depend on whether when you're walking, you spend most of your time like a flamingo with one leg up, or you spend most of your time with 2 legs down, as you're required to do if you're an Olympic walker. Okay, so which of these is it? And I'm going to argue, and they're based not on this evidence, but on some other evidence, that it's actually kinesin spends most of its time with one of the legs up, so it's more like this rather than this. But both of these are basically the same. Both of these have the property that they move along a single protofilament. And in fact, Jeff Gellis had some very nice evidence early on suggesting that kinesin didn't walk over at random over the microtubule lattice because the bead that moves in an exact beeline. The problem with that experiment, it's not a problem, but that experiment doesn't distinguish between whether you're walking astride 2 adjacent microtubules or walking on a single one. We now believe that it is, in fact, walking on a single protofilament because of this business of always taking 8 nanometer steps no matter what. So I'm going to move on with the topic and ask one of the deepest questions that one can ask in mechanochemistry, which is how is the chemistry and how are the chemistry and the mechanics actually coupled? This was touched on in the questions before. There's an intimate connection. It's not possible to change the chemistry without changing the mechanics, and vice versa. There's a reciprocal relationship between these two, as there must be under physics. But one of the questions that's been long asked is how is the ATPase cycle, which you have ATP being hydrolyzed to ADP and phosphate and then release and then nothing, how is that cycle somehow coupled to the mechanical motion of this motor, which makes at least one of the heads move, and we know they move in strict alternation. And if you're going to have 2 heads moving in strict alternation, there's this question of why don't you lift both your legs at once and float away? If I'm walking down the street and I lift both my legs, I fall flat on my face. Well, so I don't float away because of gravity, but that's a technical detail. Kinesin has this problem. How does it maintain this processivity? I told you before it moves about 100 steps before floating away on average. So that means it very successfully negotiates this kind of a cycle before it actually lets both heads go and enters into these states. So you want to suppress these rates into these steps while enhancing all of these. And there are 3 places in the cycle, this is broadly construed, where you need to do this. This is true for all processive motors. It's true for dyneins, myosins, kinesins. The heads have to coordinate. They somehow have to talk to each other, if you want to use that kind of language. If you're in this situation, the role of the front head is that you don't want to come off. You want to wait for the rear head to step because you're trying to step to the right. In this situation, you have the front head. You want to prevent that from unbinding or you come loose. And you want to take the rear head, and this is going to turn out to be important. You want to keep that from rebinding because then you could just start chewing up fuel to no good end. You wind up with what are called futile hydrolyses. Then you get around to this situation, and now, of course, what you want to do is enhance the binding in the forward binding site. That's the step itself. You want to enhance that. You want to promote forward binding. And meanwhile, you don't want to let go with the rear head, because if you did that, once again, you'd be floating in the breeze. So you have these 3 places in the cycle where so-called gates can occur. Gate in the sense that one of the myosin or kinesin or dynein heads has to somehow pause or connect or communicate in its cycle with the contralateral head in order to keep things going on an even keel. You have a central pattern generator in your brain which tells you when to walk with your left leg and when to walk with your right leg. That's why you don't lift both your legs at the same time. Kinesin does not have a brain, I think. So how does it do this? And in a short talk like this, I can only begin to touch on the subject, which is vast, but I want to give you the flavor for how this works. And so it requires me to take you actually through a mechanochemical cycle. So in its barest, simplest form, stripped bare of all other things, very trivially, you have a molecule which is poised in some state. You're going to bind ATP, in this case, onto the front head. It's going to cause a physical change in the molecule, a conformational change, which is going to result in a step. This is widely believed to be a so-called neck-linker docking for the experts of the audience, but that doesn't really matter. The point is it throws this head forward, this head then binds, and then you do it all over again. And that's the cycle in its barest forms. And those are the 3 places that I talked about before where you have to invoke these gates. And the first question I want to ask relates to so-called gate A. And the one way to think about it is kinesin a chauffeur or a backseat driver? Or in other words, in a chauffeur situation, it's the person in the rear is controlling what the person in the front is trying to do. No, that's the other way around. I said it backwards. The backseat driver is when the rear person is trying to do it. Chauffeur is the person in the front is conducting the enterprise and taking it where you're supposed to go. So does the rear head tell the front head what to do? That's this. Or does the front head control the situation? And so the role of gate A is to prevent this premature ATP binding, and there's actually quite a literature on the topic. Why, for example, don't you bind ATP prematurely to this head, which then leads to an ATP hydrolysis, which then could lead to this thing floating away? What suppresses this? There are basically 2 things that get you going back up here. One, which is the possibility that the front head, when it binds ATP, actually pulls the rear head off and regenerates this state. And the other possibility is that when you have this this chemical structure, ADP and phosphate on the rear head, or just ADP on the rear head, that prevents premature binding of ATP in the first place. So either you don't bind the ATP, or you do bind the ATP but rip the rear head off. And there have been two schools of thought. There are a series of papers from this school, a series of papers from this school, all providing evidence for this. And in fact, these two ideas aren't actually mutually exclusive, so they can both coexist. And probably something like that really is going on. But there's actually excellent evidence that when this head is bound, ATP can't get on there for various steric reasons. And so that prevents this or suppresses this activity, and that's what keeps gate A going. But what about gate B? All right, well, now you want to prevent not premature ATP binding, you want to prevent premature ADP release, in particular from back here. So the thing loads on, and this is a bad thing if this happens. If the rear head can bind back on again, then of course what can happen is you can lose that. And if you lose that, then an ATP could come on, you'd hydrolyze, so you could regenerate the state. That's great, but you haven't gone anywhere. So now you've just chewed up an ATP for no particular good reason. Or worse yet, God forbid, another ATP could come on the other situation. Now you got your heads reversed, and then you go float away. So either you're going to lose processivity by floating away, or you're going to chew up an ATP for no good reason. This is all bad. You need to suppress that. So there's a big argument in the field about what really makes this happen. Does one of the partner heads actually dock on its contralateral partner and keep that from happening? Can you evoke, once again, strain between the two heads to do this? And without going into a lot of details, the questions really come down to, is the rear head in this state actually bound or unbound? Is it really the way I've drawn it here with this thing floating in the air, or is it actually with the two heads bound all the time? And that gets us to the question of what is the configuration that kinesin is in when it's waiting to step? And you can get into that configuration by starving the system for ATP. You starve the system for ATP, it's sitting there, where's my ATP, where's my ATP? And if it's truly the flamingo, it'll go up into that one-head bound state. If it's not, it'll keep the two heads bound. And different people have different results. So here's one lab, says one head's bound. Okay, that's good. Here's 3 more papers with some of the same people as on that paper, actually, saying, no, it's two heads bound. Here's a group in England that says, well, they're bound, but they're bound in a funny way. They're bound to each other and parked in this dock configuration. And then here's us, and of course, this is the right answer. There is one head bound, but that head is actually mobile, and that mobility accounts for actually the reason why you see these kinds of data. So I'm not going to have time to go into the reasons for all of this. I'm simply going to summarily dismiss my competition using PowerPoint. It's a great tool. All right, so I want to tell you about some actual experiments that point to this. If you're truly in a flamingo position, then if I somehow lengthened your leg, then you could get back into that two-head bound position. And a number of groups have done experiments to try to lengthen that leg to get that two-head bound state, because that two-head bound state gives you direct predictions about things that ought to happen. And I'm not going to go into all of them, but I'm going to point out that at least 3 groups, including my own, have gone to this neck linker and added a few amino acids. These 6 in particular here, and Ahmed Yildiz, working with Ron Vale, put many others. He put 6 all the way up to 24 into this position. David Hackney put in some of these positions, and they did a number of experiments and interpreted them. We would interpret their experiments in a somewhat different way. We managed to get a whole lot of data on this. We probably have the largest physiological data set on single molecule kinesins ever published, which I'm about to present to you. And what we did is we inserted these 6 amino acids to produce a double head bound state, and boy, did this get weird. So here's what got weird about it. This kinesin, when you watch it move, actually does the usual thing. It moves forwards, in nice 8 nanometer steps, and occasionally takes back steps. Back steps have been observed before in kinesin. Maybe somewhere between 1% and 3% of all steps in kinesin actually go backwards. This one steps backwards maybe 5% of the time, but it depends on the load, of course. You load it a lot. If you load kinesin down a lot, basically it stalls out and stops moving, and what happens is it stays stalled for a few moments, and then both heads let go and it comes off. You lose processivity. This mutant doesn't do that. This mutant does something really weird. As you increase the force towards stall, it just sits there oscillating back and forth, forward and backward stepping with equal proclivity, getting nowhere on the average. And now you go to a so-called superstall force, so you're pulling it backwards, and this thing is now walking backwards. Okay? Now, we don't believe it's synthesizing ATP, but it is walking backwards, and it does so processively. So it does so processively over distances of a micron or more. The same kind of distance the kinesin moves forward, this one can move forward or backwards. This is just saying that. These are two different mutants. These are the kinds of distributions of distances they run. And what you're supposed to notice is that all three of those are pretty much the same value. So yeah, so they run forward to the same direction. This one, which is the wild type, can't go backwards. But these guys, if you look at the backward distribution, it looks about the same. We can actually measure how fast you go backwards as a function, for example, of the concentration of ATP, and that saturates. The data like this are part of the reason I was telling you that we don't think it's synthesizing ATP. And the backward step is very rigorously that same 8.15 nanometers is the crystallographic distance. So that fits. And by the way, these data have been fit by a model that I'll tell you about in a moment. Now, for the statistic nuts among you, it's going to turn out we're going to need to invoke a particular property, which is a Markov characteristic, which is we're going to ask, if you step forward, do you care when you step forward whether the last step you took was backwards or forwards? Is there any correlation between what you did last and what you're doing right now? And the answer is no, and you can test that in a number of ways. This is you can compare, for example, the simultaneous probability of a step pair, like 2 forward steps in a row, or here 1 forward step and 1 backward step, and you can compare that with the probability of these statistically independently, and that ratio should be 1 if these things have no memory of what they're doing, and they do that. And this is important because it turns out that this property is consistent with a common biochemical starting point to start either the step cycle or the back step cycle. And so it shall be. There will be a common starting point. So here's the Kinesin force-velocity relationship, so you can measure how fast it goes as a function of load. So here we're loading it more and more and more and more. And of course, if we load it to the point where it stops, that's the stall force. And that stall force depends on ATP, so we can measure that. And that's the point of stall. And unlike regular kinesin, which comes down to stall and floats away, this one actually passes smoothly right through that line and then goes backwards. And for the physicists among you, doesn't that look like a diode characteristic? OK. We can measure the ratio of forward to back steps. The point at which the ratio of forward to back steps is 1 is the point where you're getting nowhere. And so that's also stall. And the stall that we get from by this metric is the same as the stall we get by the other metric, which is nice. That means everything's self-consistent. But the ratio of forward to back steps actually depends on the ATP ratio. Without going into a lot of detail, in a great number of biochemical models for how kinesin works, it turns out that the ratio of forward or backward steps would not depend in quite the same way on the ATP ratio. And so this actually constrains the number of the models we could look at to a very few number. So that's good. It helps us to model. Here, again, fit by the same model as before. Again, all these red fits are fits to a single model. Here we're looking at something called the randomness. This is basically a second-order characteristic of the motion. It's about the variance in the motion. It's not how you go, but how far what are the excursions from the mean on the average before you get back. This is the actual mathematical expression from this. It looks like something like the mean square displacement divided by the average distance, and it's normalized by the distance. So this has a value of 1 when you're a Poisson stepper. So if every time a Geiger counter clicks, you take a step. That would give you a randomness of 1. This is the reciprocal of that, so the reciprocal randomness of 1 is also 1. And real kinesin has a randomness of about 1, which means actually that it's acting like a nice Poisson stepper. This thing does some funny stuff, and actually, since it goes backwards, the denominator becomes negative, and so you actually have, for the first time ever, a negative randomness. And again, this is fit by the model. And it stalls out. So these are the data that I already showed you, so everything we know about this can be summarized in a series of biochemical steps. We can even go beyond that, because although these data were used to fit the model, there's one more thing that we measured that we didn't use to fit the model, and that's the classic thing you always do with kinesin, the so-called Michaelis Menten curve. By the way, there are 8 curves here, 73 data points, 10 free parameters, and 63 degrees of freedom. Okay, here we go. So this is how fast you go as a function of ATP concentration. This is the Bayes' so-called Michaelis Menten characteristic. It's actually not quite Michaelis Menten because of the complications of the chemistry, but it comes a very close approximation of that. And these data happen to fit pretty well to that, even though they weren't even used in the fit. And so that gives us boosts our confidence that this is the sort of thing that's going on. So I'm not going to take you through all the gory details here, except to point out that that this is, to the best of my knowledge, the first true two-headed kinesin pathway. In the past, there are lots of publications which talk about the cycle for one head. This is the cycle for both heads. This includes all the gating and all the everything, and this includes the possibility that kinesin can step forwards, can step backwards, and can hydrolyze ATP in a futile way. We've left out, for simplicity, parts of the biochemical cycle that are too fast to measure and therefore are not rate limiting in any of these things, so we don't care about them so much. For example, phosphate release and ADP release occur relatively quickly and more or less the same place. So you don't actually see phosphate coming off as a separate step. Many of the myosin diagrams that will turn out to be important, you'll see it, but not here. Okay, so to take you through it very quickly, to step in the forward direction, you go from 1 to 2 to 3, and then we've unwrapped this, so you're back to 1 again. So you want to start the cycle here. That's the flamingo state, the ADP waiting state. One leg is off the ground, and the first thing that happens, ATP binds to the front head, which doesn't have anything on it. And when ATP binds, that causes presumably the neck linker to dock or something that causes the rear head to get thrown forward. It gets thrown forward, loses its ADP, comes off, and then you regenerate the flamingo state, and you're right back here, and that's a forward step. Okay. Backward steps, it turns out, can proceed by 2 different pathways. The fact that they proceed by 2 different pathways is the thing that gives rise to that ATP dependence of those 3 things. If we didn't have this, if we didn't have those 2 pathways, you can actually show that they wouldn't have that characteristic. And so there's the rearward step, and you can get rearward in 2 ways. You can either start by stepping forward to point 3 here, then go backwards, and then bind the head backwards. So that would be 1, 2, 3, skip to 4. So 1, 2, 3, and then you skip to the 4th state. So what happens from here is you lose the ATP from the rear head. When you lose the ATP from the rear head, you generate a state that looks like it has got nothing in it. And if you've got nothing in it and you now bind ATP to the front head, and then not to the front head, and then ATP to the rear head, and you go back to the flamingo state, you've now walked backwards without doing any good. And you can also hydrolyze ATP in a futile way. I will leave the details out in the interest of time, but the reason why we like this pathway is that we can fit it. We can fit all the association and dissociation constants we need. We can fit the characteristic distances for the steps. We get numbers. We can check a number of those numbers actually against transient flow kinetics. So my colleague Stephen Rosenfeld, then at Columbia, now at Cleveland Clinic, has actually measured using modern fluorescence methods, stop flow transient flow kinetics, some of these rates, and in the cases where we can measure the same rate, or what we think is the same rate, we're actually coming up with very, very similar numbers. So this is another confidence builder that this is sensible. You have a characteristic so-called distance parameter for a step. That's a number which turns out has to be smaller than the physical 8 nanometers. That could be a distance to transition state. If any of these numbers came out to be 8 nanometers or larger, we'd be very worried. That one is not, so we're not worried. This one is not. It's negative because that's the back step, and we're not worried. So by all the sanity checks, this fits. And here it is in a nutshell. I'm almost done. We can understand the pathway basically in terms of forward stepping here. And this is just the size of the errors basically gives you the normal rates under conditions of relatively weak load and saturating ATP. So under those conditions, you get a lot of forward steps. You don't get a whole lot of back steps. This is not broadly exercised. You do get some futile hydrolysis. There's the other back step pathway. There's the futile hydrolysis, and you actually get quite a bit of that. And then you can actually do the dissociation, which we haven't modeled in here, but that's the step where it would occur. You can go change the conditions, because now we know all the rate constants. It's just a matter of changing the mathematics. And now at very high loads, you begin to see lots more back steps and lots more futile hydrolyses. And rather than take you through all those diagrams, it's much easier to see it this way. Here at saturating ATP, this motor at low load basically is stepping forward most of the time. That's the blue curve. It steps backwards almost never, but it does chew up a lot of ATP. Now, the wild-type kinesin plotted on this would do much better. It steps forward almost all the time, chews up ATP for no good reason, almost never, and almost never backsteps. So real kinesin, not this mutant, does even better. But this is pretty good performance. Why aren't all kinesins built this way? Well, you can see that because if you change now the ATP to a lower concentration, you see what happens is these curves drop dramatically, and now all of a sudden you only have a 50% chance of stepping forward. You spend a lot of time chewing up ATP for no good reason, and you do a lot of backward stepping. So this motor is very poorly efficient. It has the same processivity as kinesin. It goes the same distance. It goes 1.4, in this case, microns, every time you give it a microtubule. But it doesn't do very well with fuel economy. It's not a Prius. All right, in conclusion, this was a seminar at many levels. You know, I told you about the very beginning stuff, and I told you about the latest stuff. The conclusions from the latest stuff are that we can actually write down what we think is a minimal mechanochemical pathway for two-headed kinesin. To solve this, we had to use 5 states. Our data are completely consistent with fluorescence results, which I didn't show you, that can be measured in bulk biochemistry. The reason why this flamingo state is relieved is we lengthen the leg, and so it has reduced innerhead tension. But despite the fact that it reduced innerhead tension, it still does gating. It still moves processively. It still does all the things that real kinesin does. It just pays a price in terms of low efficiency. And because gating is retained in this situation, it therefore follows that the front head is not pulling the rear head off, because if you make the leg longer, so it can't pull it off, it still does its thing. Therefore, there's something else going on, and we think it's actually the physical topology, the disposition of the molecule. You can use a lot of words to describe this, the linker orientation per se, and not the tension that's actually responsible for the gating. And so, and that gating is significant. ATP hydrolysis is actually almost 10-fold faster in the rear head, and ATP unbinding is 6-fold faster in the front head. So it's a remarkable motor. It's only a few nanometers across, but it's keeping your brains active through my seminar, for which I thank it. And I thank you. So I acknowledge my collaborator, Steve Rosenfeld, who did the transient flow kinetics, and most of this work was the work of Basin Clancy and Joan Andreasen, and Basin is now off in the private sector. He got scared because there aren't enough grants in academia these days. All right. Questions? Answers?
Okay, hi. There are no dianines in plants. Therefore, kinesin is going in both directions. So have you looked at a kinesin from plants that go in the opposite direction?
The short answer is no. There are kinesin families, as you know, that do go in the opposite direction. These are the C-terminal kinesins. CAR3, NCD are examples of kinesins that go the opposite direction on a microtubule. What we do know about those 2 particular kinesins is that they are largely non-processive. So they're like the myosins Jim Spudich talked about. They have a very short duty cycle. They come down, they give a little power stroke, and then they float away. And so you can't watch them drag cargo anywhere. And presumably the only way they could work to carry a vesicle, for example, would be in an ensemble, so that you have enough of them so it's like the itsy bitsy spider with one of the legs down at all times. And so that's the presumption of everyone in the field. But it's equally possible that someone will discover a processive -n-directed kinesin in the future. You saw that vast tree of possibilities, and experimentally people have only begun to scratch the surface. I can tell you I haven't looked. I'd be very loath to say, has anyone looked? I mean, I don't have that kind of knowledge.
I don't
think anybody's published on it.
I have a couple of questions. They are kind of related.
Sure.
The first one is for the one-head bond model. And does your stepping trace indicate large fluctuation when the head is up in the air, and the next moment this position fluctuation is smaller?
Okay, so if I understand the question, in a situation in which one of the heads is bound, are there going to be large fluctuations in the other head?
That's true.
We've actually done that experiment and published it, and the answer is yes. You can show that the head is free because you might remember one of my graphs, I showed a little DNA molecule connected to a bead.
I didn't see it.
Okay, well, you didn't see that one, but some of you saw that one. It took 16 nanometer steps. I didn't have time in a short seminar like this to talk about the whole rest of that experiment. The whole rest of that experiment involved taking that bead and seeing if we could wiggle the free head forward and backwards using the optical trap. And could we get it to swing forward? Could we get it to swing back? And the answer is yes, you can. Not only can you get it to swing forward, but you can actually see it swing backwards a little bit to bind to the binding site. So you can get lots of information out of that, and I'd refer you to Block and Gaida's paper, which will take you through that in gory detail.
Now the second question. It's about the based on the Paul Selman's work. I think, if I understand correctly, the reason they were able to measure the step size of the kinesin is because every moment this head is bonded to the microtubule.
So their interpretation of their experiment is that they had a two heads bound state at all times.
Yeah.
We don't agree with that interpretation. We think they're incorrect. But you would get their same data, it turns out, even if that were not the case.
Is it because of ATP concentration?
No, because, well, yeah, basically. They were working at extremely low ATP concentrations. And so the molecule just settled into whatever shape it was in, and they measured it. And so they're basically got the shutter open the whole time. And so they're measuring the average position. So the average position is here, and then it moves, the average position is there. And their experiment simply doesn't distinguish whether both heads are bound or not. So that was part of their analysis, but it's not actually part of their data set. And you don't actually have to make that assumption in order to analyze their data. I'm not questioning their conclusion. I think it's a correct conclusion, that there is hand-over-hand motion, and they correctly reported that, but it does not require that both heads be bound at all times, which was a lemma they used, which they didn't have to.
Thank you.
Thank you, Steve. That was great. In the interest of time, I'm going to give you my questions at the reception, but I just want to point out that your little bouncing ball is like using ATP, right? So.
No. That bouncing ball is an optically trapped sphere. This is the Black Lab logo. And,
I see.
And so it's doing diffusion. And then we captured it, and we're suppressing thermal motion.
I understand it completely.
Ah, okay.
So, good. So we're going to close now with Sam Reck-Peterson, who will sum up and provide some insights.
All right. I'd like to start by thanking all of the speakers today. It's been a tremendous day. So I personally have been fascinated by molecular motors for my entire scientific career so far. So today, not as brief as you may think. So this day has been really, really fun for me, and I hope you've all enjoyed it as much. I want to spend just a couple of minutes in closing talking about some things that I saw as challenges for the future that were brought up by the speakers, and also some common themes that I thought came up throughout the talks today. So I think the talks today really spanned the field beautifully, from trying to understand how single individual biological molecules work to trying to build synthetic motors or even in silico motors, to trying to understand how, when motors don't work properly, how that causes disease. So what are some of the challenges for the future? I'm just going to mention three that I think came up throughout the day today. I think the first is that, particularly from Steve's talk, is that there's still a lot to learn in regards to how individual biological motors work, but still for kinesins and different types of kinesins, for myosins and many different types of myosins, and then for the really bizarre motor that we work on called dynein. And so there, really, ultimately, the question is, how do these motors use chemical energy to create force that moves things around inside our cells? And I think that, as you heard from people today, we can really be inspired by learning how biological motors work to build better and better synthetic motors. Which brings me, I think, to the second challenge for the future, which is to continue to build better and better synthetic machines that are more sophisticated and programmable, and ultimately with the goal of being able to control them for therapeutic and engineering purposes. And then the third challenge, I think, for the future, which was also pointed out by many of the speakers today, is to understand how motors, biological motors, are regulated inside our cells to perform so many different functions. And it's clear from the talks today that biological motors, when they don't work properly, can cause disease. And I think that in the future, we're going to, by studying biological motors, make great strides in understanding and hopefully treating neurodegenerative diseases, cardiomyopathies, and ciliopathies. I also wanted to highlight two common themes that I saw running throughout the talks today that are not unique to the study of molecules, motors, and movement, but I think are particularly important in this field. So the first is the importance of developing new technology. So of course, new technology is important in all scientific endeavors, but I think in the motors field in particular, often our desire to understand how molecular motors work has really led people to come up with new technologies to study them. And I think that surely this is going to be important in the future. And finally, sort of the last common theme that I saw as I was sitting listening to the talks today was the incredible importance of collaboration. So we heard talks today that had, all of which had collaborations between many different types of scientists, including mathematicians and physicists and chemists and engineers, biologists, clinicians. And I think this aspect of studying motors is one of the things that really drew me to the field, is the ability to work with people from different disciplines to tackle a really complicated and fascinating problem. And so I think that it's events like this symposium, which do bring together people from many different fields, that can be a catalyst for collaboration, as well as really inspiring for people like me. So thank you again. I want to particularly thank some of the people that were involved in organizing this event. First of all, Roz Siegel, whose idea this was, and she really brought a lot of people together to plan this event today. I'd also like to thank the Radcliffe Institute, in particular Becky Wasserman, also Maura Madden from Academic Ventures, Phyllis Strimling and her team from Academic Events Management. Also the organizing committee, which includes, in addition to Roz, Elizabeth Engel, Joan Ruderman, Dimitar Sasolov, Tom Schwartz, and Judith Steen. And with that, there will be a reception downstairs, so just back down where the posters and lunches were, and the posters will be up for a bit longer as well. Thanks again.